Hi guys, we are starting unit 2 of the semester today. So chapter 4 is the first chapter of the second unit. Chapter 4 is about transport mechanisms that occur across the cell/plasma membrane of cells. Um, so we're going to go over specifically the different types of transport mechanisms. So transport mechanisms that require energy and transport mechanisms that do not require energy. So to briefly introduce the gist for the lecture, there are passive transport mechanism and passive transport mechanism. There are three examples, three types: simple diffusion, facilitated diffusion, and the third type is osmosis. So passive transport mechanism do not require cellular energy. Right? So passive transport mechanisms do not require cellular energy. And when we say cellular energy, in this context, we are referring to ATP. So you can say loosely ATP or cellular energy. In this context, they mean the same thing. So all passive transport mechanisms do not require energy. And the other one, the active transport mechanism, always require, you guessed it, cellular energy, CE/ATP, okay? So active transport mechanisms require cellular energy or require ATP. Passive transport mechanisms do not require cellular energy or do not require ATP. Now, why did I split osmosis from passive transport? There's a reason to that. So here is the reason. When we talk about active transport mechanism across the cell membrane, we have two types of cargoes. So both on top here, we are talking about transporting solutes across the cell membrane. Right, so when the cell transports solutes, it can use simple diffusion, facilitated diffusion if it relies on passive transport mechanism. It can use primary active transport, secondary active transport, or bulk transport when it requires, ATP. In both cases, we are talking about solutes being transported across the plasma membrane. But when we talk about osmosis, it's unique. Osmosis, we are talking about the transport of the solvent across the cell membrane. solvent, solvent versus solute. And in this case, for cells, the solvent is water, H2O. The best solvent there is for life, right? H2O is the solvent. Solutes, anything else besides water. So when you are transporting water across the cell membrane, when you're transporting water across the plasma membrane, the process is called osmosis. And when you're transporting solutes, then you can have all other mechanisms. That's why I split osmosis from simple diffusion and facilitated diffusion. Because simple diffusion, facilitated diffusion, we're talking about transporting solutes. When we transport water, it's a different name. Okay? So that's the gist for the lecture for chapter 4. So without further ado, let's get the ball rolling with the first slide. So we talk about, the first one is simple diffusion. Again, simple diffusion is an example of a passive transport mechanism. So passive transport mechanism, simple diffusion. So the first of three we're going to go over. Right? So the three examples of passive transport mechanism, we're going to go over the first one, simple diffusion. And since it's passive, ATP is not required. Cellular energy is not required. So if there's no energy requirement, the next question is, what is the incentive? What is the engine? What is the motor driving this passive transport mechanism? Because in nature, nothing happens for free. There must be a reason. There must be an incentive. And that reason, that incentive, driving passive transport mechanism, in this case, driving simple diffusion, the incentive is called concentration gradient, delta C. So when we talk about gradient, it's always, it goes from high concentration gradient and it moves to low concentration gradient, right? So always, when we talk about a gradient, it's always from high going to low. So this is high concentration here. It's moving to another compartment, lower concentration. That's why you see here the arrow moving from the site number 1. It moves to site number 2. Driven by concentration gradient. And ultimately, when equilibrium is reached, like when site 1 and site 2 have the same concentration, then there's no more net flux. In other words, the net flux, NF, equals 0. What do I mean by net flux? So in other words, for every molecule that moves from site 1 to site 2, there's another molecule that will move from site 2 back to site 1. If there's 2 from site 1 to site 2, then there's 2 from site 2 to site 1. So at equilibrium, net flux equals 0. And that happens when both sides have the same concentration. Half a molar on site 1, half a molar on site 2. And in that case, you have a net flux equals to 0. But over here, when you have site 1 at 1 molar and site 2 at 0, then you have a huge net flux from site 1 to site 2. And once site 2 starts to accumulate a concentration, so the gradient now is less, right? So there's less gradient, so the net flux slightly decreases. That's why you have a smaller size arrow over here compared to this thicker arrow over here. So the concentration gradient will dictate the net flux. The stronger the gradient, the higher the gradient, the stronger the flux. The smaller the gradient, the smaller the flux. And ultimately at equilibrium, when there's no more gradient, then the flux equals 0. And so that's the concept of simple diffusion. Now let's add some details into this process. Right. So remember, we're talking about transport across the cell membrane. And we learned previously that the cell membrane is basically built from phospholipids, right? So the cell membrane / plasma membrane, this is based on phospholipids. So this is basically fat, so lipid base. More specifically, mostly phospholipids, right? So in order for a solute to move across the membrane through simple diffusion, the solute has to be fat-soluble solute. So simple diffusion transports fat soluble solutes, meaning solutes, chemicals that are compatible with lipids. So they simply allowed to move across the membrane and driven only by concentration gradients of these solutes. Right, so the concentration gradient of the fat-soluble solutes drive their transport across the membrane. Since they're compatible with fat, they can move through the membrane. So because of that, you don't need proteins at the membrane to facilitate their translocation, right? So just with only the phospholipid bilayer, if you have a chemical that is fat soluble, that chemical will move through them across the membrane, driven only by the concentration gradient of that particular fat soluble chemical. Okay, so simple diffusion transports fat soluble solutes across the cell membrane, across the plasma membrane. And the incentive, the driving force, is concentration gradient. Now, what if the solutes are not fat soluble? What if the solutes are water soluble? Which brings us to the next slide. Right? So here, it addresses that question. So if you have hydrophilic solutes, so let's break that word down, hydrophilic. Hydro means water, and philic means loving, right? So in other words, hydrophilic solutes this word hydrophilic means water soluble soluble solutes. In other words, these chemicals, these water-soluble chemicals, will not be allowed to cross the cell membrane, will not be allowed to cross the plasma membrane, will not be allowed to cross the phospholipid bilayer. So they need help to be able to cross that fat-based membrane. And the assistants are transmembrane proteins that are located at the membrane, so transmembrane proteins. And these transmembrane proteins are required now to help transport water-soluble solutes. And these are small solutes, small size, like electrolytes. So when we're talking about small water-soluble solutes, we're talking about sodium. So these are cations, sodium, potassium, electrolytes, right? Calcium. This is a divalent cation, 2+. We also not only positively charged cations, but we also can have negatively charged anions like chloride, Cl-. So these are examples of small water-soluble solutes. These cannot cross the membrane, since the membrane is made of lipid-based macromolecules, specifically phospholipids, right? So they need help. And these channel proteins, these transmembrane proteins are actually channels. They have a pore, P-O-R-E. They have a channel. They have a passageway right here. So this here, this funnel, this passageway opens up, and then cations can flow through it, right? Can flow through here to get in. For example, sodium. Sodium can get in, right? Same thing, calcium can get in. But in the case of potassium, it's the opposite. Potassium is, instead of getting in, they're getting out. So potassium is leaving. And chloride also is coming in. I should have drawn that before. So here, take that out. Take this out. And redraw here in. Chloride is moving into the cell. And all that is depends on the concentration gradient again. Because of the concentration gradient, these electrolytes are shown in the direction I just drew earlier. Okay, so they're small size cargo that would fit in the channel, in the pore. So that's a pore, an opening, P-O-R-E, pore, or a passageway inside the protein. These are transmembrane protein. When they open up, there's a hole that basically opens up, and both the outside world, this is outside the cell, and this is inside the cell, becomes continuous, and then the electrolytes can move in or out depending on the concentration gradient. Now the next question is, what if the solutes is water soluble, right? But they're not small. They're not small like electrolytes. What if they're bigger than the pore? So that passageway, it's limited in terms of size, right? What if we have large? So what if we have large hydrophilic molecules? For example, glucose. The glucose would be too big for that pore, so it cannot go in. So glucose is also water soluble, so it cannot go through the membrane. And this is when it goes in here, it cannot go in because it's too big, right? So glucose, too big for the pore. It won't go in through the channel. What happens? The answer to that is on the next slide. So we have another type of transmembrane protein that are dedicated to transport large size water-soluble solutes, large size water-soluble cargo, such as a glucose molecule, which is significantly larger because it's a compound larger than simple electrolytes like a sodium, potassium, chloride, calcium, right? So on the next slide, and here, in this case here, we call it facilitated diffusion. So let me write it down somewhere. This is facilitated diffusion. It's not simple diffusion because we require transmembrane protein. Okay, so facilitated diffusion. Facilitated, facilitated diffusion, diffusion. And the first type of facilitated diffusion is through transmembrane protein. They are channels for small size hydrophilic solutes. Yeah. And on the next slide, we're going to deal with the large size hydrophilic solutes. I'm getting ahead of myself, so we're still under facilitated diffusion, specific ionic species. So transmembrane protein are required. So you have here, these are transmembrane protein. This is a protein here. So this is a complete protein working as an ion channel like that. So this showing you its structures. So from the beginning of protein and the amino terminus, the beginning of the protein, and the carboxyl terminus, the end of the protein. So you have the beginning here and the end of the protein here, going through the membrane multiple times. And when they come together in terms of the tertiary structure, they have an opening here. So they can shut the opening or they can open up the opening. So when they shut the opening, nothing fluxes through, nothing goes through. But when they open up, you see the pore right here. You can see inside the cell. So that opening, this is where small size water soluble solutes will go through that hole, right? That opening, that channel, that passageway, to connect, to go from outside of the cell to inside the cell or vice versa, leaving the cell, moving from the inside to outside, depending on their respective concentration gradient. So again, the beauty about this is channels are selective to the specific ionic species. For example, there are channels that allow cation to flux and channels that allow anion to flux. Anion means negatively charged and cation means positively charged. Cation and ion, right? And like I've said earlier, transmembrane protein are required. And this is an example of facilitated diffusion that I wrote earlier. Now that's for small size water-soluble solutes. I repeat, this type of facilitated diffusion is specifically for small size water-soluble solutes. Moving on to the next one, it should be addressing large size water-soluble solutes. So this is dealing with large hydrophilic molecules, transporting large hydrophilic molecules across the cell membrane or the plasma membrane. EG here stands for example, so glucose. So glucose is it's too large for the channel. It wouldn't go through the pore. So we need another type of transmembrane protein. And this transmembrane protein, we call it a carrier protein, specifically glucose carrier protein. So this is another type. The carrier protein is not a channel. It does not have a pore. It does not have a passageway. It does not open up. So, you can't see through a carrier protein. You can see through a channel, but not a carrier protein. So when glucose binds to the carrier changes its shape. This three-dimensional structure changes in a way that now it shuts the outer region and it opens up the inner region, allowing glucose to get into the cell. So this is how a glucose carrier transport glucose across the cell membrane, driven by concentration gradient. So this is outside the cell. This is inside the cell, right? So glucose will move down its concentration gradient. So the carrier will basically translocate glucose from the outside to the inside of the cell. This is how our cell obtains glucose, through a carrier protein, not a channel, right? Since glucose is water soluble, it needs a transmembrane protein at the membrane to help them translocate across the membrane. Because since it's water soluble, it won't be allowed to move through the membrane itself. It needs an assistant. It needs the assistance of transmembrane protein, in this case specifically a carrier protein, not a channel. And that carrier protein will help glucose to move in the cell across the plasma membrane. It is still under facilitated diffusion. It is still driven by concentration gradient. The only difference is the transmembrane protein is not a channel. The transmembrane protein is a carrier protein. Okay, so here, in addition to solute size, what differentiate carrier protein from channel protein? I mentioned that earlier. Channel protein has a pore. Carrier proteins do not have a pore. Carrier protein do not have a passageway. Channel protein do have a passageway, do have, they look like a cylinder, right? So when they open up, there's a continuous passageway that connects the outside of the cell to the inside of the cell. Carrier protein do not have such passageway, do not have such pores. Now moving on to active transport. Now all active transport mechanisms require transmembrane protein. All active transport mechanisms always require cellular energy / ATP. An active transport mechanism is much more selective than passive transport mechanisms. It has a stronger say, if you will, of what comes in and what goes out of the cell across the plasma membrane. And the driving force here on the active transport mechanism, the driving force is cellular energy. The driving force is ATP. So since the driving force is energy, active transport mechanism can allow the solute to go against its concentration gradient, meaning it is possible for a solute to move from a high concentration, the opposite, from the low concentration into a higher concentration area, right? Passive transport mechanism is driven by concentration gradient, meaning it is always moving from high concentration to low concentration. But under active transport, since active transport is not driven by concentration gradient, active transport mechanisms are driven by cellular energy / ATP. They can move a solute against the solute's concentration gradient, meaning they can transport that solute from a low concentration into a high concentration, right? So that's what I meant here when I say it can go against concentration gradient. And exquisitely selective. And ATP, or cellular energy, its driving force. And there's always a transmembrane protein involved. Okay? TMP, transmembrane protein. So that's the definition for active transport mechanism. So comparing and contrasting active transport from passive transport. So both passive transport and active transport are transport mechanisms moving solutes across the plasma membrane. Passive transport does not require energy. Passive transport mechanisms are driven by concentration gradient of the solutes being transported. Active transport mechanisms always require cellular energy, always require transmembrane protein at the cell membrane. And the driving force here is ATP or cellular energy. An active transport mechanism can go against the concentration gradient of the solute being transported across the plasma membrane. So let's go over specific examples of active transport mechanism, beginning with the primary active transport mechanism. The reason what we call primary, we also have a secondary active transport mechanism. So here, one of the most important transmembrane protein involved in active transport mechanism, we call it the sodium potassium ATPase, or the sodium potassium pump. These transmembrane proteins are so important, they have been conserved through evolution. They are present not only in human cells, they are found across the entire domain of life called eukarya, meaning eukaryotic cells have displayed this sodium potassium pumps. And when we move to the prokaryotic domain, they have something similar to this sodium potassium ATPase. It's not identical, but yet the process is so important that the concept is very similar. So this is again another evidence that all living organisms share a common ancestry on this planet. And this is shown by the conservation of this sodium potassium ATPase, sodium potassium pump. This is not only a transmembrane protein, this is not only a pump, but it is also an enzyme, meaning it has enzymatic activity. That's why it says ATPase. ASE here stands for an enzyme, and ATPase means this transmembrane protein can hydrolyze ATP, can break down one covalent bond and the ATP molecule to produce ADP and energy. And that energy that is released is harvested by the transmembrane protein to translocate, to kick out 3 sodium and to bring back inside the cell 2 potassium. That's why we say sodium potassium pump. So every time that the ATPase hydrolyzes 1 ATP, it has enough energy to kick out 3 sodium and to bring in 2 potassium, right? So 3 sodium is being expelled and 2 potassium is being brought inside the cell. So the cell in this case is losing 3 sodium, but gaining 2 potassium when this pump consumes 1 ATP molecule. So this is called primary active transport. And again, the sodium potassium pump, this is an example of a transmembrane protein. Another type of transmembrane protein, right? So the protein is located specifically at the cell membrane. So that protein is coded by a gene, goes through gene expression, and then shipped specifically to the membrane. And it sits there, right? And its function is to consume ATP, to kick out 3-sodium, and to bring back in 2-potassium. And we're going to learn this ATPase throughout the entire semester. So it's going to come back throughout the semester. You'll see it. You can appreciate it from the nervous system all the way to the digestive system, all the way to the urinary system. So the organ system that control all other organ system, the nervous system, or the organ system that handles, cellular waste, the urinary system, or the organ system that allows us to absorb nutrients, the digestive system. This sodium potassium pumps is everywhere. It is involved to set up a way for the cell to simply absorb, for example, glucose, right? So this is a tremendous transmembrane protein having one of the most important function. And some cells use up to half of all of its energy, almost half of all the cellular energy, almost half of the ATP molecule at the disposal to maintain this homeostasis of sodium concentration by using this pump. So this pump in some cells, for example, in our kidneys, spend half of all the energy to basically to maintain this sodium, this important electrolyte. So this ATPase is a very, very expensive transmembrane protein. It consumes ATP to basically expel 3-sodium, bring back in 2-potassium. And that's an example of primary active transport. The next type of transport is called a secondary active transport. This is a little bit more abstract than the primary active transport, because the secondary active transport is actually being set up. It always requires the primary active transport. So the primary active transport sets up the conditions that is used by the secondary active transport. And in the case of secondary active transport, you can transport 2 different types of cargoes, 2 different types of solutes. In this case here, for example, this secondary active transport is transporting glucose, one type of cargo. Simultaneously, it is also transporting sodium, right? So this is sodium. And if the 2 solutes goes in the same direction, we call it co-transport, or symport. So you can say co-transport or symport up here, right? So the solute goes in the same direction. Glucose goes in, sodium goes in, or it can go out. So if the solute, both of them go out, it is also symport. So it has to be the same direction. So you can either go in, all the solute go in, or both solutes go out, that's symport. If they go in opposite direction like here, right? So this is in, going in, but then this is going out. Then you have counter transport. So you have here counter transport or antiport or exchange. Here, this is opposite direction. Up here, this is same direction. So the solute moves same direction, then we have symport. If the solid moves opposite direction, then we have antiport. So that's the basic definition. Now let's go over how does it work, right? Unfortunately, this part here, your book does not does justice to this beautiful mechanism, because the book, maybe for simplicity, does not show you the requirement for this to work. So in other words, you need to, this is showing you step 2. Step 1 has been, omitted by the book. So imagine there's a sodium potassium ATPase right here. And this is requiring ATP that we learn. And what's going to happen is the ATPase kicks out 3 sodium, and it brings in 2 potassium. That's your sodium potassium ATPase. So this consumes ATP, right? So by doing this, what's going to happen is the cell is setting up a concentration gradient of sodium. So there's going to be very little sodium concentration inside the cell, because the ATPase, the sodium potassium ATPase, is kicking out sodium, and it's making a lot of sodium outside of the cell. So it's setting up this concentration gradient of sodium. And that concentration gradient helps the sodium, this one, to use this transporter to get in. So when sodium gets in by this sodium glucose transporter, glucose hits a free ride. So here, this here is free ride for glucose. Right? So this step here requires the concentration gradient of sodium. And that concentration gradient of sodium was set up by the sodium potassium ATPase. Again, one more time. The sodium potassium ATPase consumes energy to set up a sodium concentration gradient of sodium across the cell membrane. That concentration gradient of sodium uses the sodium glucose transporter or sodium glucose symporter to bring sodium back into the cell. When that symporter, sodium glucose symporter, brings sodium in, it also brings glucose in. So glucose just, gets a free ride along with that sodium through the sodium glucose transporter. So this is how cells obtain glucose in the process. So, and this is important, guys. This step is critical, and here's why it's critical. When sodium gets in, this is driven by concentration gradient. So this step, this particular step here, no energy is required, right? So here is no cellular energy or ATP is required to drive this step. This step is purely driven by the concentration gradient of sodium. So sodium simply goes down its concentration gradient, right? So this transporter, sodium glucose transporter, transports sodium down its concentration gradient. But glucose gets a free ride. Not only that, glucose goes against its concentration gradient. Inside the cell, the concentration of glucose is higher than outside, yet glucose still gets in because that glucose is required by cells. So this step here is, there's no cellular energy required, but this step will not happen if ATP is present. So ATP consumes energy to set up the sodium concentration gradient of sodium. Outside is large, inside is small. So that step, the primary active transport mediated by the sodium potassium ATPase, consuming energy to set up the sodium concentration gradient across the cell membrane, driving sodium back in by using the sodium glucose symporter. Glucose gets in free of charge. So that's an example of a secondary active transport. In this case, co-transport, because sodium and glucose both goes in the same direction. Both are going into the cell. Now, the panel B on the right side shows you the same concept, but now we are talking about the two solutes moving in opposite direction. And same thing here. This step is driven by the concentration gradient of sodium. And this concentration gradient of sodium was again set up by the sodium potassium ATPase over here, right? Same thing, sodium gets out, potassium gets in. So again, 3 sodium, and the sodium potassium pumps use energy to bring in 2 potassium. So you have here a small sodium concentration inside the cell, a humongous sodium concentration outside the cell. Okay, so that gradient is set at the expense of ATP consumption by the sodium potassium pump. And once that concentration is set up, sodium gets back inside the cell through this sodium proton pump, right? And this is an anti-port, meaning sodium gets in, but potassium gets out. Not potassium, proton gets out. So the cell it's bringing in sodium, but it also allows the cell to exclude, to excrete, to get rid of the proton. And this mechanism comes back in the urinary system. You know, when you're going to see here how cells are basically excreting free floating protons to get rid of the acids, metabolic acids, cellular acids, waste acid coming from your cells. This is a powerful, powerful mechanism that will come back later in the semester, okay? So both of them are secondary active transport. Both of them relies on the sodium potassium ATPase or the sodium potassium pumps to set up the concentration gradient of sodium across the cell membrane. Once that concentration is set, in this case, sodium gets through the sodium glucose symporter, allowing the cell to absorb glucose. In the second case, sodium goes inside the cell through sodium proton antiporter. In other words, the cell obtains sodium, allowing the cell also to excrete, to lose protons. So that's secondary active transport. Moving on to osmosis, passive transport. In this case here, moving back to passive transport, but now we're talking about transporting a solvent across the membrane and not the solute. So again, the same thing. If it's a passive transport mechanism, it is driven by concentration gradient. In this case, the concentration of water, right? So passive transport, no energy required. Water moving across the membrane, driven by concentration gradient of water. Again, meaning high concentration, moving down to low concentration. Right? So, for example, here we have much more water on the outside, less water on the inside in terms of concentration. So water just moves in. Over here, the concentration of water is higher on the inside than on the outside. Water moves down its concentration gradient. And the same similar concept, you can say that wherever salt goes, water follows. So water always go into a compartment where there's more solute, there's more salt. Okay? So you can say either way. The water goes down its concentration gradient, meaning water always move from a compartment where it has higher concentration of water toward a compartment where there's less water in terms of concentration. Or water moves into a compartment where it has more salt concentration, more solute concentration. Now, the concept of osmolarity, this is to have a way to express the concentration of particles in solution. And what it means is these solute, for example, you have some solute that are not dissociated in water. So glucose stays as the same. So you have 1 molar of glucose is the same as 1 osmolar of glucose. But some solute, for example, sodium chloride, they dissociate in water. So let's say 1 molar of sodium chloride, it becomes 2 osmol because there are 2 particles here now contributing to lower the concentration of water. So you have NaCl. When you put them in water, they dissociate into Na plus Cl minus, right? So you have here the Na here, it's surrounded by a hydration shell. And the same thing with the chloride, it is also surrounded by hydration shell of water. So that positive charge is attracted to the negativity on the oxygen molecule. because oxygen has a higher electronegativity than the protons. So you have the two protons here have a delta plus, the positive dipole, it's called in chemistry. Whereas the negative charge here is attracted to the hydrogen of the water because the hydrogen has a positive dipole of the water molecule. So the negative charge on the chloride is surrounded by the positive ends of the water molecule from the protons, whereas the positive charge on the sodium is surrounded by the negative dipole on the oxygen. We call that the hydration shell. Because of that, when you put sodium chloride, sodium crystals in water, they split into two particles, each of which contributes to its own concentration, lowering the concentrated water. That's why 1 molar of sodium chloride becomes 2 osmol of sodium chloride. Glucose cannot dissociate like this in water. So the 1 molar of glucose is the same as 1 osmolar. So in the case of glucose, the concept of molarity and osmolarity is the same. But when you have salts that can dissociate in water, then molarity and osmolarity is no longer the same. And this becomes important because if you think about it, here the purple solute is impermeable, meaning this is the membrane here. So the purple solute is not allowed to leave the cell, right? It cannot cross over. But urea is allowed to move in. In that case, so what's going to happen is the particle, the osmolarity becomes important because that can basically dictate the water of the motion of water. You know, so for example, if you have a solution, 3 molar, so in other words, a solution containing both glucose and sodium chloride, right? So if you have that solution, so you have 1 molar solution. So in that solution, you have 1 molar glucose and 1 molar sodium chloride. When you put that in solution, it becomes 3 osmol because you have 1 osmol from glucose, 1 osmol from Na plus, and 1 osmol from Cl minus. Right, so that's the concept between molarity and osmolarity. Okay. So when we have solute that can split, then we have the concept of osmolarity. If we have a solute that cannot split in water, then osmolarity and molarity is the same. Let me erase this before I move on to the next slide. So this showing you the concept again. Osmotic pressure is an indirect way of illustrating the concept of a osmolarity. So you have a membrane here. And what is important to realize is these solute molecules cannot cross over. So the membrane is not permeant to the solutes. Only water can flow. Because of that, we call this semi-permeable membrane. It only allows water to move across, not solute particles. So the solid particles are not allowed to move across from side 1 to side 2 or vice versa. So what it means is side 2 has more solid particles. Side 2 will also have a higher pressure, higher osmotic pressure. Water will flow from a low osmotic pressure to a high osmotic pressure. You're going to say, whoa, 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 whoa. The gradient definition is high to low. It's true. Water is moving from a higher water concentration to a lower water concentration. So it's not violating anything. But in the process of doing this, you can see that water moves from a low pressure compartment to a higher pressure compartment. So the high osmotic here has a higher attractive force water moving in. So it can basically have a stronger suction. It sucks water in. It takes water from side 1 and it translocates water into its side 2. We call it concept osmotic pressure. So it is due to the concentration of solute, the gradient, the concentration gradient of this impermeable solute across a membrane. And that can be expressed as giving rise to a pressure onto the membrane, right? So it creates a pressure on the membrane. The solute particle creates a pressure, we call that osmotic pressure. And that pressure attracts water from a higher concentration water to a lower concentration water. So in other words, the water is trying to decrease the concentration on side 2. So ultimately what happened is the water content on side 2 will go up and the water content on side 1 will go down because side 1 will lose water, side 2 will gain water to the point where the concentration on side 1 and concentration side 2 becomes equal, then the water flow will stop. At equilibrium, there's no more net water flow, right? So solid concentration, 300 to 500. Now the solid cannot move, only water can move. So water will move from high concentration to a lower concentration. So water can move because the membrane is not allowing the solute to move. Tonicity. It's the same concept. So if you have a semi-permeable membrane, there are some solute that cannot leave the cell, whereas water can move in. So the concept, it's called, if you have an isotonic solution means just inside the cell, the solution inside the cell has the same solid concentration as the outside the cell. So if you put a cell in an isotonic solution, the volume of the cell will not change. So if you have a beaker here, and you put the cell here, and you have the water. So if you have a hypertonic solution, right? So the solution is hypertonic, meaning there's more salt on the outside. What's going to happen is the cell will lose the water. Water will move out. Wherever salt goes, water follows. So in that case, the cell will shrink. So if you put the cell in a hypertonic solution, the cell will lose water. Water will leave the cell, go into this extracellular solution. In the process, when the cell loses water, the cell shrinks and it will die. What happens to the opposite condition where you put the cell in a hypotonic solution? If that's the case, hypotonic means the solution contains less salt than the cell, so water will go in. Again, water goes wherever salt goes. In the hypotonic solution, inside the cell contains more salt than outside, so water will go into the cell will gain water, the cell will swell, and ultimately the cell will burst. Okay? So that's why I wrote here, hypertonic solution strings the cell, hypotonic solution swells the cell. Both cases, the cell will die. Let me go back on the active transport. Need some space. Okay, let's use this space here, this slide for active transport. For active transport, we talk about primary active transport and secondary active transport. There's also another transport. I mentioned a vesicular transport somewhere at the beginning. This one. Okay, let's use this page then. This vesicular transport is also active transport. This vesicular transport is also called bulk transport. BULK. And under vesicular transport, we have exocytosis, exocytosis means when the cell needs to export something. Exocytosis, like a neuron when it exports neurotransmitters, right, through exocytosis. So neurons, when they release neurotransmitters, more specifically, when axon terminals of neurons release their neurotransmitters, these axon terminals relies on exocytosis. This is an active transport mechanism. It requires cellular energy. It requires ATP. Okay, so when the cell exports some type of specific cargoes, and it uses exocytosis. The opposite is called endocytosis. When the cell imports something, endocytosis. And under endocytosis, there are specifically 3 types of endocytosis. There's a pinocytosis. Pino, P-I-N-O, and the word cytosis. One word, pinocytosis. There's phagocytosis. Phago and the word cytosis. Pino, P-I-N-O, cytosis. Phago, P-H-A-G-O, cytosis, one word. Pinocytosis is when the cell is known as cell drinking. When the cell brings in a large volume of water containing dissolved electrolytes, right? So when the cell is importing a lot of solvent containing solutes, we call that cell drinking. Phagocytosis is known as cell eating. When the cell is importing, for example, the entire bacterium, the entire bacterial cell, like a macrophage, when it eats up the entire bacterial cell, it relies on phagocytosis. When the cell brings in, for example, a viral particle or a virus, that's through phagocytosis, cell eating. So both cases, we are talking about a humongous cargo. This is large size imports, right? And the last one, the most expensive one, this is transporting VIPs, if you will, like when the cell is transporting in insulin, right? This is a very important chemical messenger used by the endocrine system that we call hormones. Insulin helps body cells to absorb glucose. So it doesn't matter how much glucose you have floating in your vascular bed, in your blood vessels. If you don't have insulin, the cells cannot absorb glucose. So the cells need to bring in insulin so insulin can allow the cell to absorb glucose. And when insulin shows up at the front door of our systemic cells, of our body cells, the body cells bring this VIP in. It brings this insulin inside themselves, and that mechanism is called receptor-mediated endocytosis. The most expensive, the most selective transport mechanism of all. The receptor-mediated endocytosis. The word receptor, receptor hyphen mediated, mediated. So receptor-mediated, the word endocytosis. The most exquisitely specific, the extremely expensive, but that's for transporting only extremely important cargoes, extremely important chemicals. In this case, a very important chemical messenger, insulin. And on this note, I'm going to end here chapter 4. And in the meantime, study well, stay safe, be healthy, be happy. Until next time, take care.